Good afternoon, ladies and gentlemen, and a warm welcome to the Sustainable Urban Agriculture Forum. My name is Visalini, and I will be your MC for this plenary session. The focus of track 1 is on sustainable solutions for crop quality, crop diversity, and crop health. We will be hearing from our distinguished speakers on crop development and cultivation techniques and the potential of agri-food business. The push to strengthen the agri-food industry through the use of technology is further compounded by the COVID-19 pandemic, when food resilience becomes a highly critical agenda. We have a lineup of 4 speakers today. Without further ado, may I invite Dr. Deivanai Subramanian, research scientist of RP, School of Applied Science, to share about the development of dwarf tomato as a superior crop for indoor farming. Dr. Deivanai, please.
Yeah, thank you, Visalini. So my presentation is organized in 4 subunits. So it consists of introduction and objectives, optimization of growth conditions, evaluation of hybrids, and key features. So in the introductions, let me discuss about the need for the new tomato varieties for indoor conditions. So greenhouse technology all the way has attracted the attention of growers worldwide in the last decades, and several farmers have successfully grown the leafy greens in the indoor environment. At RP, we are exploring the possibilities of introducing tomatoes as an alternate crop for indoor cultivations. The reason why we have chosen tomato is because it is a widely grown vegetable crops worldwide, and a majority of present day cultivars are indeterminate, and they continue to grow for a period of 2 to 3 years and requires unique cultural practices, which in turn is labor intensive. Therefore, we felt that developing tomato types with an altered plant architect would therefore be ideal for indoor conditions. So we have put up few objectives to achieve the goals. So our objectives consist of optimizing the growth conditions to effect hybridization, to analyze the genetic variations of fruit size, shape, and yield, and to evaluate the hybrids for reduced plant height. So we practice conventional breeding techniques, and the breeding techniques is shown here. The scheme is shown here briefly.
Hi, Dr. Deivanai. Sorry to interrupt. Yep. Can you please share your screen? Thank you.
Oh, I'm sorry. Okay, the title of my presentation is Development of Dwarf Tomato for Superior Crop for Indoor Farming. So the presentation has been classified into 4 subunits, which consist of introduction and objectives, optimization of growth conditions, evaluation of hybrids, and finally the key features of the hybrids. So in the introductions, let me briefly describe the need for the tomato varieties for indoor conditions, because greenhouse technology has all the way attracted the attention of the growers worldwide, and several growers are successfully now growing leafy greens. At RP, we are exploring the possibility for introducing tomato as an alternate crop for indoor conditions. The reason for choosing tomato is because it is a widely grown vegetable crops, and majority of tomatoes, indoor tomatoes are indeterminate that can grow for 2 to 3 years, and it requires a unique cultural practice, which in turn is labor intensive. So therefore, we felt that developing a new tomato type with altered plant architect would be appropriate for indoor conditions. So to achieve this objective, we have optimized the growing conditions to effect hybridization, to analyze the genetic variations, especially for fruit shapes and numbers, and finally to evaluate the hybrids for reduced plant height. So here we followed conventional breeding techniques. The breeding techniques is shown in this slide. Briefly, the selection of the parental type is at most important. So once the parental line is chosen, then have to practice hybridization. And after the hybrids usually combines the genetic makeup of both the parents, and rarely the hybrids excel the parental performance. In such cases, the hybrid seeds has been straight away released as sun hybrids. But most of the cases, the hybrids were intermediate between the parental lines. So screening have to be followed from second generation of the hybrid, and it has to be continued up to sixth generations. Only thereafter, evaluations for specific trait have to be carried out. So basically, the breeding methods will have setting and objectives, and then we have to look for the genetic variations, then followed by selection of desirable trait at subsequent generations. Selection starts at the F2 generation of the hybrid, and it has to be continued up to sixth generations of the hybrid, and every hybrid generations testing need to be continued, and finally have to be evaluated for the specific trait. So the objectives in general for any breeding program starts with the increasing the yield, or also breeding for tolerance towards biotic and abiotic conditions, photo insensitivity, and finally improved quality. So in our breeding programs, we selected locally available tomato types, and then it has been grown in different environments to verify the fruit sets. In all the environments, the plants were able to flower and produce, able to flowers, but unable to produce fruit set. It might be due to the variations in the light intensity and also high humidity. So we alter the growing conditions in the growth chamber. So here we can see the growth chambers filled with fruit set. So using these conditions, we hybridize the plants. The hybridization scheme is shown here. So using this hybridization procedures, we have generated few hybrids. The hybrids were evaluated along with the parents. So in general, the evaluations was focused on two different areas. The first phase, we focus on increasing the yield of the hybrids, and the second phase was reducing the plant height. In every evaluations, we focus on assessing the genetic variations as well as the mode of gene actions. There are three different types of gene actions which consist of additive gene actions. If the trait of interest is governed by additive gene, so that characteristics can be easily fixed within the cycle of selections. If it is governed by either dominant gene or interactive gene, so we need to postpone the selections. So that's why, so if the selections is postponed, then we have to look for the inheritance of the trait to assess the percentage of gene contribution to the characteristics. So here we have used 15 different characteristics broadly related to fruit maturity or fruiting period, fruit size, and finally the fruit yield. So the data was subjected to test the adequacy to see that whether they follow the normal distributions. So here most of the trait studies follow the normal distributions, hence the grouping of parents were followed. So here the clustering analysis shows that the closeness of the clusters indicate the strong relativity of the genetic content, while the distance clusters shows the diverseness of the parent. So with this parental informations, the hybridized products were evaluated. So these are all the comparison of the hybrids with that of the parents. So here, most of the hybrids were just intermediate between the parental performances. So none of the hybrids outperformed the parents. So we have forwarded the hybrid seeds for screening subsequent generations. So here are some examples of these selected hybrids. So at first generations, you can see the combination of genetic material from both the parents in the hybrids. But in the subsequent generations, for example, if it is in the second generations, you can see a lot of genetic diversity. So selection starts from the second generations of the hybrid. So we focus on the fruit size, the numbers, as well as the sweetness for the selections. So here we give weightage to total weight, total soluble sugars, and also the fruit numbers. So two hybrid perform much better in the second generations. So we chosen that for developing high yield plant type. For evaluating the hybrid, for evaluation of reduced plant height, we approach three strategies. One is introducing the seedlings to the low light conditions. Yeah, another one is applying the hormones, especially some growth hormones, either by treating the seeds or at the time of vegetative phases, one foliar space was given. And the third method, we use backcross breeding. So in all these methods, the seed sample, seeds were the leaf samples were collected, and biochemical parameters like plant pigments, especially chlorophyll B and carotenoids, were estimated along with the free radicals. Here, the picture shows the superoxide dismutase. Higher estimates of chlorophyll B content as well as the carotenoids and low values of free radicals were taken as a criterion for the selection of the hybrids. So based on this information, the hybrids has been forwarded and they were grown in the greenhouse conditions. So now we are evaluating the fourth generations hybrids. So so almost all the hybrids shows the reduced plant height. So in general, the selected hybrids found to be earlier in durations, and they have higher fruit sets. So they shows reduced plant height with moderate sweetness, and most of them are highly tolerant towards low light conditions. So in terms of benefits, the generated hybrids are high yielders. They can grow much faster than the parental lines, and the fruits are quite different from the parental lines, and it looks like a teardrop or a pear shapes. So our focus of selections was laid only to the novel shape and more numbers as well as the sweetness. So this varieties, once we have evaluated it in 2 more generations, it will be ready for growing in either in vertical forms or in hydroponic cultures. So if anybody interested, they can also use our material to multiply the seeds. So that's the features of our newly developed dwarf tomatoes. So the study is still in progress. We are evaluating the dwarf plants. So our team member consists of Dr. Ritu Bala, Dr. Ventris De Souza, and Ms. Chun Fongi. So thank you very much.